Hello, my name is Dr. Roderick L. Roll, and today I will be talking to you about carbon. Carbon is a very diverse atom. So let's look at the diversity. What we will need is our periodic chart. If we look at the periodic chart, we see that carbon is in a certain row and it is in a certain column. So this brings us back to these terms. Valence shell is 2. So therefore carbon is in row number 2. This means the outer shell is the second shell. Also, carbon is in column number 4. It's a Roman numeral 4. So therefore it means that in this outer shell, which is number 2, you will have 4 electrons. If every atom is trying to achieve the noble gas arrangement, this means you would take 8 and subtract it by 4. This gives us a bonding capacity of 4. So the valence of carbon is 4. All living things are composed of carbon. So therefore, most science majors, health science majors especially, would take a class called organic chemistry, which specifically looks at molecules that have a carbon backbone. In this image, we have a planetary model of carbon. You should also know how to draw the Lewis dot structure. So for carbon, all you need to know is the valence electron. Since there are 4 electrons in the outermost shell, you draw the C and 4 dots around the C to represent a Lewis dot structure. The simplest organic compounds are called hydrocarbons. These are nothing more than molecules that have carbons and hydrogens linked together covalently. The simplest of the hydrocarbons is called methane. In the image below, we have several structures of methane. I would like for you to now draw the Lewis dot structure for methane. This is what the structure should look like. Make sure you're familiar with drawing Lewis dot structures of simple hydrocarbons. These are several variations of hydrocarbons. Remember, hydrocarbons are composed of hydrogen and carbon. So the first box, we have a carbon skeleton of varying lengths. The first one has 2 carbons and the second one has 3. So hydrocarbons can have varying lengths based on the carbon number. The second box is showing a branched form versus a unbranched form. When we talk about sugars in the body, such as starch, we would look at different branched versus unbranched forms of starch. The next image shows us when we add a double bond between carbons, we can change these sugars such as glucose and fructose. And then finally we'll talk about a fat, which is called a steroid. It will form these ring-like structures. So these are variations of hydrocarbons that we will see when we get to the chapter where we talk about macromolecules. Larger hydrocarbons will be the main molecules found in molecules such as gasoline or fat molecules. Now we will talk about functional groups. Once we have created our hydrocarbons, we can add what are called functional groups. These are atoms that will be attached to these molecules. These atoms will affect the function of the molecule. It will also affect the shape, and it can affect the chemical reactions. Here are a list of several functional groups that you will see in this class. We have the hydroxyl groups, which will be OHs. We have the carbonyl groups. This would be that double bonded oxygen linked to a carbon. We have our carboxyl group, COOH, or you will see it written as COO. Then we will talk about the amino group, which is NH2 or NH3. Then we will talk about the sulfhydryl group, sulfur with a hydrogen. Then we will look at a phosphate group, oxygen, a phosphorus, and 3 oxygens. And then we will finally conclude with the methyl group, CH3. The first one we'll look at is the hydroxyl group, the OH group. This will be seen in molecules such as alcohols and sugars. The next functional group is the carbonyl group. This will be seen in sugars. So you take a hydrocarbon chain, you add a carbonyl group, and it turns it into a sugar. The next functional group is a carboxyl group. You take a functional group, you add a carboxyl group to it, and it can turn it into an amino acid. So down in the bottom of the image, I have circled a word. If you look above that word, you see that the carboxyl group is COOH. And to the right of that, you only have COO, and the hydrogen was released. This means that if a molecule has a carboxyl group, that molecule has acidic properties. Why? Because it can release hydrogen into the solution, therefore driving down the pH. The amino group has the opposite effect of the carboxyl group. If you look at the red circle in the image, you see that we have an NH2 become an NH3. What happened is this property of the amino group can accept hydrogen. So therefore, a molecule with an amino group has basic properties. The next functional group is the sulfhydryl group. One of the 20 amino acids that we will have to learn is called cysteine. Cysteine has a sulfur group. So if you look at that cysteine amino acid, you notice that on the bottom of the chiral carbon, you have the amino group, NH2. On the top of the chiral carbon, you have the carboxyl group. And to the right of the chiral carbon, you have what is called the R group, or the variable group. In that variable group, you have a sulfur. That sulfur is used to create a disulfide bridge. So in the formation of the tertiary structure of proteins, when a linear amino acid sequence folds on itself, it can be held together by ionic bonds, disulfide bridges, hydrogen bonds, and so on. One of those bonds is a disulfide bridge, which is held when 2 cysteine amino acids are adjacent to one another, and they will form this bridge. The next functional group is a phosphate group. The phosphate group will be seen when we discuss metabolism. So the energy chapter, the energy molecule is ATP, adenosine triphosphate. it has 3 of these phosphate groups attached. Finally, we will talk about the methyl group. The last chapter in this class, we're going to talk about a concept called epigenetics. And this is when we will see how these methyl groups can modify transcription and translation. So the functional groups can make molecules become even more. So you can take a simple hydrocarbon and turn it into a sugar. You can take a simple hydrocarbon and turn it into an alcohol. You can take a simple hydrocarbon and turn it into amino acids just by adding functional groups. So we are going to now look at some molecules that were formed when we add the functional groups. These molecules can form variations of themselves, and we call them isomers. We will discuss 3 types of isomers: structural isomers, geometric isomers, and enantiomers. The first isomer we'll talk about is type 1. So type 1 variation is called a structural isomer. This molecule's isomer differs in its covalent partner. So if you look at our image, we have a glucose molecule in its linear form and a fructose molecule in its linear form. If we added up the carbons, the oxygens, and the hydrogens, we have the same number, C6H12O6. The only difference is the position of the carbonyl group. In glucose, the carbonyl group is on the end of the molecule. In fructose, the carbonyl group is within the molecule. So that's the only difference. So all we did was move the carbonyl group, so it has a different covalent partner. So in glucose, the covalent partner for the carbonyl group is carbon 2. In fructose, the covalent partner for the carbonyl group is carbon 1 and 3. So again, the carbonyl group has a different covalent partner. This is an example of structural isomers. How does that matter, or why does it matter? Well, that structural isomer, fructose, actually tastes sweeter to the taste buds. So if you look at this chart here, and we were to compare, let's look at a pear versus a carrot. Which of the two would taste sweeter? Well, obviously, pear tastes sweeter, but how? If you look at the concentration of fructose in a pear versus the concentration of fructose in a carrot, a pear has way more fructose than it does glucose. Therefore, based on the taste index, a pear would taste sweeter. Type 2 variation is called a geometric isomer or cis-trans isomer. This molecule's isomer has the same covalent partner, but it differs in its spatial arrangement. So if we look at this image below, we have the shaded portion. The shaded circles are carbons, and they are covalently linked together by a double bond. If I held the one on the left and rotated the one on the right, I can move the red circle from the bottom to the top. This rotation of 180 degree is what happens between the trans version and the cis version. All we did was change the spatial arrangement. If you change the spatial arrangement, you change the shape. Geometry is the study of shapes mathematically. So geometric is changing shapes. So the shape changes between the trans version and the cis version. Here is another example of how we can change the spatial arrangement. It has the same covalent partners, but it changes in spatial arrangement. We rotate one of the carbons by 180 degrees. How is this relevant? Well, if you look at a saturated fat versus a unsaturated fat, you notice that the unsaturated fat has that carbon double bonded to another carbon. This is going to create a kink. All right, this kink can be altered by hydrogenating this oil. In the hydrogenation process, sometimes we create a trans kink. So if you look at the difference between the cis kink and a trans kink, all we did was change the spatial arrangement. We just rotated one of those carbons by 180 degrees. Why is this important? The trans fats cause arteriosclerosis. So the trans fats are not good for you at all. If you look at this chart, you notice that yes, saturated fats are not good for you. You still have an increase in mortality when you eat saturated fats. But if you eat trans fats, you increase the mortality rate even greater. The third type is type 3. So type 3 variation is called an enantiomer. This isomer is a mirror image of itself. The example we'll look at is a drug that is used to treat Parkinson's. With the L-dopa, this is the left-handed image, versus the D-dopa, this is the right-handed image. These two drugs, one of them is biologically active and actually helps to treat Parkinson's, versus the other one is biologically inactive. This is a picture to show you an enantiomer. The left hand image versus the right hand image. They are mirror images of one another. You cannot superimpose these two images. So an enantiomer is non-superimposable, which means it cannot be placed on top of one another and be the same molecule. Let's look at two examples. The first example is an amino acid called asparagine. The isomer to the left will taste bitter, versus the isomer to the right will taste sweet. So in the creation of this amino acid, you create mirror images of itself. The left hand will be bitter and the right hand image will be sweet. Let's look at another example. This second example is a drug to treat tuberculosis. Tuberculosis is caused by Mycobacterium tuberculosis. The left-handed isomer is the actual drug, the antibiotic. The right-handed image will cause blindness. So again, these are examples of enantiomers. One version can be bad and the other version can be good. Here's another example. This is ibuprofen. Ibuprofen is a racemic mixture. This means that in this mixture you will have equal parts. You will have the left hand image versus the right hand image in equal parts. Well, for this ibuprofen, the plus image is the one that actually gives the effect versus the negative image, the one that's circled in red, it has no effect. So when you take ibuprofen, half of the content of that drug is not working, whereas the other half is working. So if you could somehow purify and only isolate the positive molecule, then that drug will be more effective. The last example is going to look at a variation of a drug. One is legal and one is illegal, based on them being mirror images of one another. So the one to the right is used as a nasal decongestant. This drug is legal and is sold at many stores, whereas the one to the left is just the mirror image, but it is illegal. You would have to serve time if you're caught with this one. This one is meth. This slide talks about other examples of enantiomers. In a nice article in the New York Times, it talks about how in the 80s, women would take different drugs while they were pregnant, and the actual drug, the enantiomer of the drug, would actually was hurting the developing fetus in their womb. And what happened is a lot of women had miscarriages, or the babies had birth defects because of enantiomers. The next thing we'll look at is how functional groups can make even more elaborate molecules. So these more elaborate molecules are called macromolecules. You've heard of them before. They are carbohydrates, lipids, proteins, and nucleic acids.